So good afternoon. I'm Ghada Al-Haj-Flayhan. I'm going to be giving the first and the last lecture of clinical trials with Dr. Mahfouz, who will be giving you most lectures. I thought what we'll do is just introduce ourselves around the class so I know who everybody is. This is a huge lecture in terms of slide set. I'm considering as a textbook where if you want to get more, you get more. If you want to study for the exam, this is not really exam material. This is giving you a flavor. The exam would be mostly derived from Dr. Mahfouz's lectures. And my last lecture would be to cap with my experience with clinical trials, which I have been, taking on as a principal investigators since the 80s. So lessons learned, and I still learn, and it's a wonderful journey. So let's start by introducing ourselves front to back, and then we'll launch in the lecture. And you are a sharp student, yes, MS.
Okay, Hassan Talib, MD, sharp student.
Rima?
Rima Kadura, MPH, EPHD.
أهلا.
Physician and MPH graduate. I'm auditing the class.
Sure. Your first name is? Mamadou. Okay.
Isra Ahmed, MSc Biochemistry.
أهلا.
And MD student, EPHD.
Arif Al-Ahmadi, MD, MPH, Palestine.
Okay, so we have maybe a little over half the class are MDs. Can you raise your hand? 1, 2, 3, 4, 5. With the auditor, you're an MD? So 4, half, wonderful. Pharmacy. And you are?
Clinical trials, right?
Okay.
And I'm veterinarian. I'm veterinarian.
Okay, and you're taking a master at the School of... In?
MPH,
Ahlen. Okay, so as I said, this is a lot of slides, but this is just to give you a flavor of what we have. And I think if you were to remember key points maybe, and these are going to be rehearsed and repeated during Dr. Mahfouz's lectures, I think these are the ones I'm going to be stopping at. And I'm sorry, I should have thought about this, Rima. I don't have slide number to say which slide we're on. Is this going to be a problem? Okay, I'm sorry. I'll try to do that for the last lecture. I just realized it as I was sitting here in front of the class. So the course objective of this 3-1 EPHD is to understand the basic concepts of clinical trial design, including bias, randomization, blocking, masking. A lot of that is going to be covered in the coming lectures. To cover the essential of trial conduct, the statistical concepts for data management, analysis, interpretation, and to demonstrate the knowledge of advanced methods used in the analysis of specific design. You will get introduced to factorial design, crossover designs, which are not as commonly used as phase 2 and 3 trials that you'll see in journals, but nevertheless you'll see few. And also acquire some of the ethical principles that guide the conduct of clinical trials and discuss the dilemma. How many people have taken an ethics course here? So, okay, and then I think as a graduate student, you are to take the Responsible Conduct of Research module on CITI, right? Have you taken that or not yet? Rima, Responsible Conduct of Research, I think.
Last year in research design.
Okay, all right, perfect. I think this year they're asking actually the certificate to be provided by the university across all master degree. So this is my understanding.
I used to have the first homework was the CITI, but now
Yeah.
since everybody's taking it.
Right, so apparently the university is informing, so even you guys, I'm sorry, but you're going to have to do CITI or CR, although you got all the principles during class. They want to homogenize it across campus. Okay, so there is a parallel course that runs with this, which is not a requirement for the MPH-MS student at the health sciences, but certainly is for the sharp student, which is 3-1A. And this is designed to really allow you to have hands-on experience writing a protocol. Those of you in FHS who are interested in auditing, of course you can, but it's not a requirement. So basically, we use the SPIRIT guiding document and walk you through week by week what it takes to write a clinical trials protocol. And this is your weekly assignment. There is no quizzes, no exam. And the nice thing about it for MDs who are seriously considering to do a clinical trial project, they are so polished by the end because every week we correct them, me and Dr. Ael, who co-teaches it with me. And so by the time you're finished, you have a very polished protocol. And we have 2 sharp master MS students who submitted it for funding and got funded because it's so well written. So if you have the time and the interest, I would encourage you to do so. The next slide is the course schedule. You have it in your syllabus. As I said, the meat is given by Dr. Ziad. I'm giving the intro and icing on the cake. Course material, of course, you have the slides. We may additionally post some lectures online on Moodle for the SHARP student, but the FHS student are more than welcome to use them. We've picked up 3 textbooks on clinical trials, clinical research, and design analysis of clinical trials that are available. And there was an online live course that was given by Harvard edX, and now it is actually a library. It's taped, it's available online. I was just there last week, and it's still available, but there is a fee. Actually, it's free now. Here you go, it's free. And if you pick that slide and click on the bottom, enrolled, you will be able to address that, attend that. It's a very nice course. I've seen it. I've actually watched most of its lecture, not all of them. I haven't seen the most recent version, though. These are the textbooks. Grading, homeworks 20%, midterm exam 35%, and final exam 45%. So what I'm going to try to do, and again, get a flavor of each one of these items, is go through what's a clinical trial? How long ago did we start having clinical trial? Why do we need clinical trial? What are the types and the phases? What are the essential elements when you write a protocol? Few touch on ethical issues, how you register a clinical trial, what are the FDA requirements, and then how negative trials impact how we practice medicine and public health. So type of clinical studies. I think this diagram, which is very old from Lancet, almost 15, 17 years ago, look at type of studies. And I think if you dichotomize in your mind saying, is this an experimental study? Did they assign an exposure? Do you intervene with an exposure or not? If the answer is yes, it's an experimental study. If it's no, of course, it's not a clinical trial, it's an observational study. And if it's an experimental study where you're actually acting and assigning an exposure, is it done randomly? And if so, it's called a randomized clinical trial. And if it's not, it's a non-randomized clinical trial. So this is for clinical trial. And then for the observational studies, I think you'll get exposed to that. You're not interfering, but you have comparisons. And it's either a comparison at one time point, and it's a cross-sectional study. It could be a comparison with a case control study design, or where the outcome is now, and then you go back to look at the exposure. Or it could be a cohort, like the Framingham cohort, the WHI cohort, where you start at one point with an exposure and you follow the outcome prospectively. So what's a clinical trial definition? And the coming 4 slides are going to be various definitions by various organizations. But I think one is enough. If you want to know more, you can read more. But they all revolve around the same theme. One, it's prospective. It looks at the effect and the value, so the efficacy, safety for drug medication, for example, or use of equipment, of your intervention against a control, and it has to be in human beings, okay? So you have to employ one or more intervention. It has to be an experiment, okay? It could be therapeutic, diagnostic, it could be a device to check hemoglobin, blood sugar, it could be anything. Check smokers, pollutants, exposure, etc. Each participant must be followed from a very well-timed defined point, time 0, randomization. These techniques are all applied to all patients in a standardized fashion. Otherwise, it doesn't meet the definition of a good randomized and control, meaning you randomly assign exposure versus control or placebo, and it's double-blind. Neither the exposed subjects nor the physician, investigator, public health person, research assistant know what the exposure is. So observational studies are not clinical trials. And then what I give you is various definitions, but as you can see, I underscored experiment, random allocation, study, control, rigorous comparison. You're looking at rate of disease, your recovery, death, and any outcome in both the study group and the control. I give you the NIH definition, again, prospective research, human subjects. And there is a very nice resource on the NIH on what are clinical trials, with some links and useful information, how you register them, some fact sheets. I don't expect you to read that. But for those who may want to have a career in clinical trials, there's a lot of resources out there, and you'll get them through the slides. So this is briefly my introduction and definition. Now let's see, oops, I stole my show. Do you know how long ago we started having clinical trials? When was the first time you think a clinical trial ever happened?
You say 500 or 5,000 BC or something.
Yeah, anybody wants to go further back? Okay, that's pretty good. I didn't realize that, but in biblical time. So biblical time is when clinical trials happen, and I won't go through that. And I had a master's student who applied. I don't know, was that you? Who in his, was that you? Who in his application, sharp application, told me when the first clinical trial was. It was impressive. Anyway, so many, many years ago. But if you want to look at documentation, the first really documented trial was in 1747. The famous scurvy. Scurvy is vitamin C deficiency. So it was called the Juice Trial, and it was conducted by James Lind and contained several elements of the trial, the control, the exposure, you know, and the forward carrying, etc. 1943, the biggest funding agency for research in the UK called MRC, Medical Research Council, did a trial of Patulin for common cold, and it was the first double-blind controlled trial we can trace. So 1943. 1946 was the first exposure with streptomycin, which was used as a drug to cure pulmonary tuberculosis, a SIL, carried out by the MRC in the UK using random numbers. Now, so the UK was really at the center stage of conduct of clinical trials, and the first person who ever, as far as we can tell, proposed randomization was Bradford Hill in 1937. And also David Rubenstein, who are the founders of clinical trial. By the way, I have to stop for a minute and say I have to acknowledge Professor Jim Ware, previous dean at the Harvard School of Public Health, for having come to Beirut and sharing some of the slides here, and you'll see them. He unfortunately died last year of cancer, but he's been a wonderful resource for our students in their protocol write-up. So this is one of his slides. So this is a clinical trial of sanocrisin in the study of pulmonary tuberculosis. And what this slide shows is actually a table where we are looking on the extreme left at radiologic assessment of response to streptomycin in patients with tuberculosis. So the first column reads considerable improvement, moderate improvement, no improvement, considerable deterioration or death on the extreme left. The second column is the streptomycin group, and the third column is the control group. And as you can see, considerable improvement was seen in 51% in the strepto and only 5% in the control. Of course, the numbers were not huge. They were 55 exposed to strepto and 52 to control, but that's really a very nice comparison. And there were other differences. Deterioration was moderate to slight in 23% of the control as opposed to 9% of the strepto. And death was 27% of the control versus 7%. There were no p-test, no t-test, no value, no statistical comparison. And that was in the BMJ in 1931. So as you may know, President Franklin Roosevelt had polio, right? And so in 1938, he established the National Foundation for Infantile Paresis. And they conducted in the mid-50s very successful funding campaigns to collect enough money in John Anders' lab to look at actually some of the treatments of polio. So the polio virus was first grown in non-neural tissue. The Salk, remember the Salk vaccine? Salk and Sabin developed the vaccine. 1954-55 was the first trial of Salk vaccine, and then the vaccine was provided free to thousands of children afterwards. So this is called the Francis Field trial. Remember I told you Francis Field is one of the fathers of clinical trial. More than 300,000, mostly volunteer, including physicians, nurses, teachers, public health officials, and community members carried out the field trial. It enrolled 1.8 million children in the first 3 grades of elementary school at 215 sites. I think this is really unbelievable considering the times and the resources then. And they did it in a double-blind fashion. 650,000 children received the vaccine, 750 the placebo, 750,000, and 430,000 as control. And they were called polio variant pioneers, and they all got certificates showing that they participated as shown in this slide. And this is a beautiful picture looking at oral vaccination at the San Antonio Auditorium, where you have a huge auditorium around the corner, and you have people lined up, I would say, you know, hundreds and hundreds, right wrapping the corner in 2 lines, very impressive children, where they gave the vaccines to the children once available. So the impact of polio vaccination, of course, was substantial on disease mortality and morbidity. I show a table here with the left column showing case incidence rates, death rates, the middle column showing the rates in 1960, and the last column showing the rates in 1970. The important thing is the incidence rate dropped from 4 in 100,000 to 0.01 in 100,000 with the intervention of the vaccine. And the death rates went from 1 in 100,000 to 0.005 per 100,000. So substantial impact on public health from a clinical trial conducted so many years ago and still impacting our health, the health of all children nowadays. So it started a long time ago, and of course, the most common and threatening disorder globally, including the MENA region, is non-communicable diseases. So meaning diabetes, cardiovascular, cancer, obesity, hypertension, respiratory disorders, they account for 2/3 of all deaths. And so pivotal clinical trials are central to the discovery of drugs that can have a substantial impact on these massive disorders. This pie diagram shows what people die of. This is actually non-communicable diseases. They account for 84% of death worldwide. And if you look at the pie for Lebanon, it's very similar. I think this is a Lebanon pie. Actually, cardiovascular disease is 45, cancer is 19, and then communicable diseases is only 7%, injury 9%, diabetes 2%, other NCDs 13%, respiratory is 5%. And what I've just flashed here at you are the acronyms for all the trials that have affected how we practice medicine today. TIMI is the large cardiovascular statin trial. Freedom, Horizon, HIP, HERS, Ruth, FLEX, FIT are all osteoporosis trial. ATTACK, I think, is a hypertension trial. So you can see these trials have shaped our lives. And this diagram here shows how many hits we got on PubMed when we looked at number of clinical trials by year. They were less than few dozens in the 60s, and they were above 25,000 registered on clinicaltrials.gov. I'm sorry, picked up by PubMed. When you go on PubMed, you can just put one keyword. It gives you a CVS file, which you can transform into a graph and graph clinical trials, and I'm sure it's much more now. So 25,000 into 12. And this is by decades, and you can see we went from 1,370 in the 60s to over 200,000 between 200 and 210. So clearly, trials are important. They have an impact on how we practice medicine and public health. But why do we need the trial itself? Why do you need a trial design? Well, here are a few thoughts, again, from the textbook. Given the uncertainty in knowledge about disease course and the huge, large variation in how people respond, unless you have a rigorous setup where intervention and control are exposed exactly to the same intervention, systematically, without biases from the investigator, the environment interfering with your intervention or therapy, you will never know the right answer, right? So this is why it's so important. They are at the heart of medical advances, and usually one conducts a clinical trials for two purposes. You want to establish the efficacy of your intervention, i.e., how impactful is your intervention in the ideal setting, namely the clinical trial setting. But ultimately, you assume that efficacy is the same as effectiveness. Effectiveness is how your drug works in actual practice, in the real world. So if I have a statin lowering LDL by 40% in a trial, it may be less than that in the real world because they may not take it. Somebody else may forget it, they may not pay for it, they may be taking a drug that interferes with its absorption. So many reasons. They may go on a diet, which makes the cholesterol worse or better, it's not the drug, so on and so forth. So the trial, the pivotal trial, the one that really gives you the package to go to the FDA or the AMEA to get approval, establish efficacy, because we consider the trial to be more or less a perfect experiment or near perfect. The effectiveness is post-phase 3, phase 4, they call them post-marketing, after the drug got to the market, everybody can buy it, and how it works. So clearly, the consequences of clinical trials are profound on practice and public health. And actually, the consequences of not doing appropriate trial will lead to malpractice. We may be using drugs, MDs may associate with that, without indication. For example, digoxin for failure was a drug given to everybody, and now it's really given to select people because it's not efficacious once the drug trials were done. Now, the next slides, 3 slides, are crucial, and I think you need to concentrate with me on what these show. So I'm going to go back to the original. And again, this is in the textbook, but Jim Ware gave it to me, but it's in your textbook. So the goal of clinical research is to conduct a study that allows us to make inferences about the universe, right? I mean, when you have a trial, whether it's a trial of few hundreds or a few thousand, usually when it gets published in big journals like the New England Journal or Lancet, you're saying this is what's going to happen to the whole world. But of course, there are some limitations to that. So ideally, the findings in the study are going to be inferred to what really happens in the universe. I mean, you can already think of reasons why this may not be. But you want to get as close to that as possible. Whoops. Okay. So, the findings in the study, first of all, must reflect the truth in the study. So, how truthful was the study to the study design? Because if the study was truthful to the design, then the findings in the study are true to the study. So this is the internal validity. Findings in the study, if the study is conducted rigorously, and we're going to talk about that throughout the course, then you can say what I find in the study really is the truth. It reflects what I did. I said I'm going to do this, I did it, and this is what. But if you say I'm going to do this, and your RA doesn't know what they're doing, then what you find is not the truth. The findings do not reflect the truth in the study. This is internal validity. And then when we go from the findings and the truth in the study, we're going to say this is our findings, this is the truth. It's going to reflect the truth in the universe. So you're drawing conclusion. So from the study to the universe is the external validity. And we're going to rehash that a lot. If I do a study, and I think I show the slides, so let me not steal the slideshow. Okay, so we have to design and implement a study with a research question first, and the design must be done in such a way that there is external validity. In other words, that the intervention I'm using, the patient population I'm addressing and targeting are really reflecting the real world of that population. And I do a study plan, and then we're going to implement it. And that implementation has to be rigorous for preserving the internal validity of the study, and you conduct the study. Once you conduct the study, you go forward, you say these are the findings in the study. And if the study was internally valid, you can infer that the findings in the study reflect the truth in the study protocol, and then the truth in the study protocol, if you have enough external validity originally taken care of, you should be able to infer to the whole universe. So the investigators have to maximize the external validity of the study through the design, through choosing the question, through the type of intervention and measurement, and through the selection of subjects. If I want to do a study, this is a perfect example of hormone replacement therapy on menopausal health, such as the WHI, and I think this trial is a textbook by itself. The NIH enrolled 16,000 women in the 2000s. Randomized half, 8,000, to estrogen progesterone and half to placebo. And they had another parallel trial in 8,000 women who did not have a uterus, so no progesterone needed, and they gave them estrogen versus placebo. A lot of issues with this trial. One is the mean age was 65. Menopausal health and menopause and hot flashes happen at 50. So what you find in a 65-year-old woman who's more likely to be obese, hyperlipidemic, hypertensive, diabetic, with hormone therapy may not be applicable to the young woman. And guess what happened? When WHI was published, and actually there was a stopping of that trial 2 years, at year 4 to 5, the first year they send letters to the subjects. Here's the ethic. We're not sure we're seeing a harm signal, but stick on with us. And then the year after, which is 4 to 5 years, they stopped it. Was monumental. All women who are 50 stopped hormone replacement therapy because they automatically, the press, the media, some physician, equated the findings in the study to a what would happen in an early postmenopause. This was 2002, 2004. Telescope front to now, 2017. And actually, guidelines, which were changed then, no HRT, no HRT, no HRT, has swung back. Because when they started digesting the results after the landmark JAMA paper, they realized that, well, age matters. And timing is everything. And there have been zillion editorials on that, as I'll show you in a few slides. So you really have to think about what question do I want to answer? What is my subject population that I want my trial to apply to? And what's the best design to do so? The other point in WHI, which is perfect, is that they use oral estrogen and oral progesterone. Well, oral estrogen, although it was the most common in the US, is more likely to cause hypertension, thrombosis, DVT, stroke, pulmonary emboli. The European are much more likely to use estrogen cream, biologically identical hormones that are estradiol, 70, and that may not be as bad. So the age of the patient matters. The intervention mattered. And so the findings of WHI, of course, I would not start HRT in a woman who's older, who's hypertensive, obese, diabetic, okay? But I still start HRT in younger women for various indications, provided they don't have contraindication, although, unfortunately, we don't have a large trial establishing the efficacy of transdermal estrogen in young women. What I do, however, is I say to these young women, well, I do know from WHI, and certainly it has advanced knowledge, that the adverse event profile is a case of breast cancer 0.08% per year after 3 years. So the actual risk, not the relative risk, is important to share with the patient. And then it's a quality of life with risk, no medication without risk. But do I have the trial to tell me what it is in a young woman? No. I'm saying, well, in an older woman who has a more disease-prone environment, this is what it is. So it's likely to be less in you, but I don't know. Okay? Any questions? So clinical trials are serious business. Seriously, in the time of thought process, design, time to develop a nice protocol. It doesn't get written over a day or two. It has to mature. And it's reiterative. In other words, if I want to answer a question, my first thing is to go to the literature and see if that question has been answered. And I should do a more or less thorough job and how to look at that literature. And then I ask my question. And then I come to design the study, and I realize my question is not very feasible once I start putting the design together. So I go back and say, is this the best question? Is this the best design? And so on and so forth. And your question, as you're writing clinical trial protocol, and as you see for those who develop clinical trial protocol, which change as you're, and we tell you, think of a question very early on. You should be thinking about it now, so once you're ready to start, you know what it is. They're expensive, millions. They may be as high as 200 to 500 per trial. They usually take years. For you to have large outcomes, you have to wait 2 to 3 years at least. It's not a week or two. They can only investigate 1, maybe 2 treatments, and you'll see that with the crossover design and the factorial design. They may not apply to most patients in practice. They may not be practical. They're very difficult to get funded. They're very time-consuming, very complex. So why bother? Well, it's the only study design that can establish causation, cause, effect. It's not an association study, because when you're doing the double-blind randomization, exposing everybody to the same drug, you would hope that the other random things that placebo or control will get exposed to will be similar, hopefully. And therefore, what you're seeing is the effect of your intervention, unless during the progress of the trial there is bias. And of course, WHI had biases. Women started bleeding. They knew they were on hormone. They had breast tenderness. They dropped out. Compliance was low. Power became lower. So there are issues to be thought about. Sometimes you say it's placebo-controlled, but at the end of the day, it ends up not being placebo-controlled because of the nature of your intervention. This is why you say identical looking, smelling the same thing tablets. So they can't guess, but they can guess sometimes from the effect on them. It has the greatest influence on clinical practice. It's the strongest evidence in evidence-based medicine, and it's required by the FDA and also the EMEA in Europe for any new drug or new devices, and it's increasingly required by payer. Payers may not pay for coverage with drugs unless it's FDA approved and established. Now this diagram looks at the quality of the evidence. On the x-axis from the left to the right is the ease of conduct. And on the y-axis from bottom to top is the strength of the evidence. And as you can see, on the right hand, on the extreme left, low ease of conduct, i.e. clinical trials or meta-analysis of clinical trials, is the highest strength of the evidence. On the extreme right, if it's a very easy study to conduct, such as case report or case series, it's going to be low evidence, right? So as you go from the right to the left, you're going from lower evidence, case report, then case series, then case controls, then cohort, then randomized trial, meta-analysis of randomized trial. Meta-analysis have their problems themselves. We won't get into that, but if it's a well-done meta-analysis with low heterogeneity, it should give you very strong evidence. Now, I got ahead of myself because I'm so passionate about WHI and lessons learned from it. This is the slide I was talking about. So in the 1990s, everybody was using hormones. There were primary prevention studies and secondary prevention studies. Boom, comes WHI 202. WHI estrogen only 204. Everybody in 202, 204 stopped hormone. The American Cardiology, the American Cancer Society, American Heart Association, Hormone Foundation, everybody said no hormone. And then in the 2010, people started revising their guidelines. So the goal of clinical research is to conduct those studies that allow us to make inferences, of course, about the universe. And then the last thing I already mentioned that I should mention is beware the impact of media on how randomized trials are accepted by the public and interpreted by the public. And for example, I can tell you for the HRT story, they, for example, wrote, even physicians didn't read the statistics, Dr. Mahfouz can tell you, a relative risk of 1.3, i.e. a 30% increased risk in breast cancer above what? Is that a 30% risk absolute for anybody who takes hormone? What's the relative risk of 1.3? Anybody? Venture, it's Friday. No big deal if it's a mistake. What's the relative risk of 1.3? What does it mean? If I say the relative risk of HRT users is 1.3, what is missing from the continuation of my sentence? Compared to whom? Thank you. So a relative risk of 1.3 in WHI combined is in the combined users versus the non-users. 1.3 means a 30% increased risk over the risk of the control. Does this mean a woman has a 30% chance of getting breast cancer? Of course not, because you have to know what the baseline risk in the population is, right? And the baseline risk multiplied by 1.3 will give you the actual risk in the exposed. You subtract the baseline risk, you get the absolute risk. This is what I'm talking about when I tell you 0.08% per year. What did the media think? 30%. Any woman in her mind would never take hormone with a 30% risk. So media has a very powerful effect, and sometimes media, you know what sells? Bad news, right? Media loves bad news. They like to make impression. Good news don't sell very well. Bad news sell. So media has been very deleterious, may I say, in some occasions, not out of being vicious, but out of first not understanding. And some physicians even thought breast cancer is a 30%. Even now, today, we have people in OBGYN who are reluctant to take the time to discuss HRT with women. So keep that in mind. So what they discovered, and again, I think I'm really taking my time on this. I should probably speed up. What they discovered, this is from a colleague, Jane Colley, a very dear colleague, co-investigator, that actually timing is everything. The timing hypothesis. In other words, if you take a woman right after menopause or premenopausally, her vessels are clean. They don't have any plaque, they don't have any cholesterol. But as they get older after the menopause with estrogen deficiency and aging and weight gain and diet changes over time, they start accumulating plaques. And so there are in vitro test tube experiments and animal experiments that show you that estrogen in the young years is good on the lipids, is good for the vessels, so the beneficial effect predominates in younger women with clean vessels. But if you go far beyond and you have 60s and 70, then estrogen becomes prothrombotic, increases coagulation, increases inflammation, and therefore the deleterious effects. So timing is everything, the timing hypothesis. So I'm going to skip that. There have been zillion publications on WHI. And as I said, the average age was 65 and does not apply to younger women. So keep that in mind. Now moving on, and we're going to really touch very quickly on these. What are the type of trials? And the FDA classifies trials into prevention trials. You know, how can we prevent disease? Screening trials, how can we screen for disease, diabetes, hypertension, hyperlipidemia? Diagnostic trials, what are the best methods to diagnose a disease or condition? Treatment trial is the one we MDs are most familiar with, testing, or even public health for vaccine. Experimental treatment or combination of treatment to affect disease. Quality of life studies becoming increasingly important, improving quality of life in cancer patients or chronic condition. And compassionate use trials, they are very particular. It's a restricted use of a non-FDA approved drug before its approval as compassionate in somebody who has terminal cancer, where there is no hope to live, but there is this promising drug. And so there may be exceptions, and the FDA will grant a one-on-one basis for compassionate use or compassionate trials in general. So there are bases of clinical trial development. Again, this is from Jim Ware, which is a telescope, right? On the left end of the telescope is drug discovery. So when the test tubes, you're testing 10,000 compounds for a specific target disease. The preclinical studies, which are typically studies in mice, rats, and animal models, 1 to 3 years. And then translational study 1, i.e. starting with phase 1, then phase 2, then phase 3, takes 3 to 5 years. Drug approval itself takes 1 to 2 years. So by the time the drug from drug discovery in the test tube hits clinical practice, it's going to be easily 10 years or so, okay? And this is again from the previous CEO of Merck, who's been back to academia and then since then founded his company, Mike Rosenblatt, who basically says we're testing 5 to 10,000 compounds at Merck over 3 to 6 years. It takes us 6 to 7 years from phase 1 to phase 3 trials. It takes us 0.5 to 2 years for approval, and then we post-market. And the cost per trial is 1 to 2 billion. So 10 to 15 years investing in that company for what they call the blockbusters, right? The statins, the osteoporosis drugs. So this gives you a perspective. Now, who's funding clinical trials? Well, unfortunately, clinical trials are funded in large part by pharma because they have a vested interest in actually investing into a drug because they're going to make many more billions after it gets approved. But it's a risky business. The NIH, so basically the private sector pharma really funds clinical trials. And they outspend the NIH, the National Institute of Health, which is the biggest funding agency for research worldwide. It's in the US. Spends a lot on basic research. They spend $29 billion on basic research. This is a few years back. This is a couple of years back. The private sector spends $65 billion on clinical research. So you can see our practice is very biased by what pharma wants us to use. And I, as an expert in vitamin D, I've done all my trials in vitamin D, and it's been a struggle because vitamin D is so cheap. Nobody wants to fund vitamin D trials. So you can get a perspective of how biased practice is, and sometimes public health policy, unless the government invests into a trial that has a major public health impact, nobody's going to invest in it, especially if it's lifestyle, smoking cessation, dietary modification. These are the best interventions for NCDs, whether we like it or not. But people don't invest in them. I sit with a patient with osteoporosis and I hammer, you have to practice, exercise. And they look at me and I said, it's a prescription. It's as important as the drug I gave you, if not more important. It's cheap, it's safe, it helps more than one thing. It helps your bone, it helps your depression, it helps your brain, it helps your blood pressure. And I really think, unfortunately, we sometimes are not allowed the luxury of educating patients, but cheap public health strategies are the best strategies. And they are going to serve lifetime, okay? So you, future moms and dads of generation, exercise and healthy diet to your children is really worthwhile. This is an aside. So conflict of interest. Okay, here's another one. We doctors, or sometimes public health people who may have stock in companies, may have a conflict in investing in the clinical trials because they may be returned, because you have stocks in that company, or even if you're a physician conducting a drug trial, you have investigator fees, you get paid, you get taken on a tour to see lectures, and so on and so forth. So conflict of interest is something you always have to keep in mind. And there was a very interesting ethicist from Hopkins, who once told me, and I was kind of not quite ready to accept it, he said, I think doctors in the clinic should tell their patients before they prescribe drugs what their conflict of interest is. I thought that was really outrageous. But in the States today, every physician has, I think there is a public website where they have to register their conflicts. So patients can actually go and seek that information and find it. So we have ways to go. So this cartoon has two snakes. One snake says, is it true you've been selling your venom to pharmaceutical companies to make anti-venom? Traitor. So no one is immune from conflict of interest as physician, and public health experts keep that in mind to stay as much away as that as possible. Okay, trials of, am I going too fast? All right, I don't know how much time I have, but I think we'll get there. Okay, we have an hour and a half, so we're at 40 minutes. I think we're okay. If there are questions, stop me. This is not supposed to be didactic. I'm trying to cover as much as possible as I can. So no questions, I move ahead. We have 2 hours. Great. Am I speaking too fast? Am I speaking too fast? No. Okay. All right. So what are the, thank you, Ziad. What are the types of clinical trials? Phase 1, establish the maximal tolerated dose. Phase 2, evaluate the biologic activity, if any, and side effects. Phase 3, establish the efficacy. Remember we said efficacy and effectiveness. This is efficacy of a drug in a relatively short time frames. Typically, clinical trials are 2 to 3 years, typically. Now, I can talk from the osteoporosis experience again. For the last 2 years, the NIH has been spending some time, energy, and money with bone investigators in the US with an initiative called the FNIH, where they're trying to find surrogate markers of osteoporosis, namely surrogate markers of fractures, as outcome that would be solid enough and acceptable enough for the FDA to approve new drugs on their basis. Why? Well, at least 2 reasons. One, clinical trials are becoming more and more expensive, and with hard outcomes and long time, almost no drug pharma is willing to go into these big experiments. Merck just lost a supposedly promising blockbuster, odanakatib, for bone 2 years ago when they discovered adverse events of cerebrovascular events, and they didn't even go forward with a folder to the FDA. So one is the time and the consumption of money. 2 is, now that we have a lot of approved drugs for many diseases, is it ethical? So there is an ethical reason. So there is a financial reason, and there is an ethical reason. Can you think of an ethical reason? I almost gave you the answer. Why wouldn't drug companies, why can't they do randomized placebo-controlled trials nowadays? Or why is it more challenging or more difficult nowadays than 20 years ago?
When there is a good standard, we should use it.
Absolutely. And the AMA already said, if you have an approved drug that works, you should use that drug as a control. Now think about it. If you have a drug that has an has established efficacy, your comparator is going to have to be really much better if it's a superiority trial, or it's going to be a non-inferiority trial, and then the question is, well, who cares? Well, would you care if you have a drug that's equally effective?
And the side effects are considerable in the previous drug.
Cheaper, less side effects. So you can see the revenue to the company, unless a lot of new people adopt it, it's not going to be very much. You have to increase the size, okay, or the other way you could do it is if you're not going to compare it to an established drug, can you think of a scenario, 2 scenarios, where you can still compare it to placebo? I gave you the answer by my intro on the FNIH initiative.
Different age, you mean?
Wonderful. Why different age? Tell me something more.
Because it has proven efficacy in like postmenopausal women. What about premenopausal women?
I wasn't thinking that way. So I'm thinking risk. Okay, I can't give a placebo to a high-risk person, right? So if I have a frail 70-year-old lady, I can't randomize her to placebo if she's going to break a hip. And I personally was approached by the calcitonin company, Novartis, 15 years ago. This is a true story. They say we don't have efficacy for calcitonin in hip fracture. We have it in vertebral fracture. We want to establish it for hip fracture. I said, really? And you're going to have a placebo? They said, yes. The type of people who sustain a hip fracture are the frail older women, right? So I said, I'm not going to be a PI on a study where I'm giving a frail older woman a placebo. So I declined being a PI, and guess what? Did we ever do the trial? Many other colleagues didn't feel comfortable, and the trial never happened. So you go to lower risk. That's one. So age, I was thinking lower risk. So rather than taking an osteoporotic woman, take an osteopenic woman, younger, less risk. But if she has less risk, that means that group has less events, and therefore your sample size goes bigger. So Merck, on their odenacatab trial, which cost them a lot of money, which didn't even go to the blockbuster drugs, this is a very sad story. Do you know how many they enrolled in that trial because they were lower risk? It could be the same age, but low risk. No risk factors, no previous fracture, no family history, whatever. Do you know how many they ended up enrolling? Take a wild guess. So the typical osteoporosis hip fracture trial would be 2,000-3,000 because the rate is 0.5 to 1% per year. So 2,000-3,000. Do you know how many they enrolled? Just take a wild guess. 16,000. 16,000 with a drug that never ended up going to the FDA. So there are ethical consideration in that. So where did I get here? I don't know where I got here. But anyway, the phases of the trial. Dr. Mahfouz is going to talk about that a lot more, but usually you do the phase 1 trials in different designs. Here I have a 3+3 design, so I test the first 3 subjects. One possibility is none of them gets a side effect, so you go to the next dose, you increase the dose, and you go back. Or one gets a side effect out of the 3, then you repeat that dose in another 3. And you have to reach a steady state in the first 3 subjects you test that you can go back and say this is the dose. In other words, if none get it, you keep increasing the dose. And basically, you want, if 1 out of 3 get it, you repeat it. If 2 out of 3 get it, you reduce. And that will ultimately give you your phase 1 drug dose, right? Okay. And you go in a reiterative process. If the dose is decreased and you have a total of 6 that have already been treated at that dose, you stop the study and you declare that the dose is the maximal tolerable dose, okay, MTD. In general, phase 1 trials contain 20 to 80 subjects. Often they're not patients with a disease, they could be control because you're looking at side effect and tolerability. And you're mostly interested in evaluating safety and identifying side effect, toxicity, and defining the maximal tolerable dose. Phase 2 is a larger drug, and it determines 100 to 300 as opposed to 20 to 80, and it determines the biological activity, the efficacy using surrogate markers. Now, we didn't get to the lower risk. It could be surrogate markers. This is where the FNIH is, right? This is what got me there. So the FNIH is saying now, okay, can we have strong enough evidence that rather than using fracture as my outcome in osteoporosis, can you use biochemical markers of bone remodeling at 1 year? Can you use BMD at 1 year and say this is good enough if there is efficacy that this drug is going to decrease fracture? Now, it's a consideration. It costs the duration, costs the cost, but what is a potential problem with that? I have a potential problem with that, and I was on the committee which was discussing that. What is a potential problem with surrogate markers? Something that will tell you whether the response as established by that marker really is the response on the outcome. What happens with surrogate trials? They're shorter, right? So when a drug is shorter, what's the dangers? Assuming the surrogate is perfect, assuming it's perfect, which is an assumption, side effects, right? And think as physician of how many drugs looked great in phase 3, post-marketing, you started seeing issues and were withdrawn from the market. So when you get that shorter, you have a trade you're doing, which is a concerning trade, okay? For example, with bisphosphonate, which have been the cornerstone, again, I go back to osteoporosis, on the market for many years, since 1995. This is 22 years. For the last 7 to 8 years, we've been talking about osteonecrosis of the jaw and atypical femoral fracture, AFF. And you know what? Media took that on, and patients stopped taking the drug. You see the drug sales in the US and UK going down. And you see fracture rate, which were going down overall, because AFF are very rare, have plateaued. They're no more going down because no one is taking the drug. I can tell you 1 out of 3 patients I talk to, هل هيدا حيهري لي نيرتي؟ هودي بيكسروا بيصير مثل ازاز العظم. This is what patients pick up. Do you know what the risk of AFF is in patients on chronic bisphosphonate 5-10 years? The risk, I wrote the bisphosphonate task force, long-term bisphosphonate, I co-chaired that task force. It's actually 1 to 10 in 100,000, okay? We actually, in our task force report, looked at how many fractures are prevented. And it would take, if we take chronic bisphosphonate for 3 to 5 years, we would prevent 130 fractures of the spine, hip, humerus, or forearm for potentially causing 1 AFF. ONJ is even less. So that risk-benefit ratio comes back. So anyway, when you cut the corners and go to shorter trials, you have the trade-off of minimizing your time to see side effects, okay? But phase 2 trials, for example, in osteoporosis, were also always bone marker trial or BMD trial. And by the way, phase 2 trials may have a dose there, a dose ranging. I mean, you think you got the dose in phase 1, you're not sure, so you may start 1 less, 1 more, and go for it to get more information. And of course, the phase 3 is the ultimate. We call them the pivotal trial. These are the trials that are done on thousands, 1,000 to 3,000. The Udana-Katib was 16,000 because they couldn't afford a placebo in the high risk to confirm efficacy on the ultimate outcome, which is the fracture. Monitor side effects, compare it sometime with standard or equivalent treatment, etc. So this is what's needed for approval. And of course, the phase 4, you know, unfortunately in the Middle East, many of us have approached by pharma saying we want to do a post-marketing trial. And we don't feel very good about it because you're not really discovering something. You're just establishing efficacy. When I've become more mature and older, I've come to realize the importance of phase 4 in terms of safety. But in terms of excitement and drug discovery, they're not the same. I mean, you're discovering something, you're learning from colleagues, you're learning the pathophysiology, you're learning physiology of how the drug works, you go to these meetings, it's hardcore science. Post-marketing is the real world, but you know what? The real world matters. So I think post-marketing trials, after all, are not so, you know, redundant and glum. Okay, and post-marketing, of course, sometimes leads to withdrawal of a drug trial. So 1 to 4 trials, type of trials, preventive, treatment, device trials, etc., palliative, compassionate. Now, what are the essential elements of a trial protocol? Whoever tells you, you can write a protocol in a couple of days, you tell them, I'm not the investigator who's going to write such protocol. Because I can tell you, no matter how smart you are, you cannot have a quality trial without time and have it mature. It really needs time. And there are elements, and I'll go over the essential elements with you. So clinical trials, all of them must have a written protocol. Why do you think we must have a written protocol? It's obvious, right? I'm asking the question, but think about it. For the interim analysis, tell me more. What do you mean? Now, you're on the right track, but tell me more. So what do you mean for the interim analysis? So you're already implying the trial has an analysis plan in it, right?
Yeah, like if once you expose the protocol, then you have to stop.
Once you what?
Expose the protocol, you have to stop at that point or something. Like you have, everything should be predefined.
Okay, everything should be predefined. Excellent. Why do you want everything? You're on the right track. Why? I want to push you more. Why do you want everything predefined?
There are ethical reasons.
Okay, what are the ethical reasons? Excellent. What are the ethical reasons?
Like if the drug is just no more working, why the people are like under trouble and they are taking this sort of
okay, so basically the protocol will say
unnecessary use of that intervention.
okay, so the protocol says if you've used the drug for a year, you have to show me results and you can't go beyond that, right? So that's one of the ethical reasons. Okay. Can somebody else or you tell me more? That's an excellent reason. Yes.
So for internal validity,
Uh-huh.
we need to make sure that every protocol is followed at each step of the clinical trial.
Excellent. We talked about validity. We hammered validity. And you have to show me each step of your protocol what you're doing, exactly how you're doing it, so people can stick to that for internal validity reasons and to be comfortable that you have a solid internally valid study. Excellent. What else? So a study usually has a primary outcome, right? And maybe 1, 2, 3 secondary and exploratory outcomes. So how do you know whether your drug or your intervention has worked? How do you in your protocol tell me? How are you going to investigate that? What do you actually do? You look at events in the placebo and in the intervention, and you compare them by a statistical test, right? And you predefine, as we said, your primary outcome. So if you weren't to write your protocol, and let's say you're doing a protocol to investigate the effect of HRT on heart attacks in young women, and your effect size is not significant, but you find a significant effect on cholesterol, you send it to the New England Journal, and you tell them, I discovered a drug that affects cholesterol. That's my primary outcome. So drug protocol, trial protocol keep you honest. Okay? You can't play. You have to predefine your outcome. You stick to them. You don't change them. Okay? And the strength of the evidence for a primary outcome is respectable. The strength of the evidence for an exploratory analysis after the fact, your data mining, it's nothing wrong, but say up front, this is exploratory. This may provide the basis for a clinical trial that may be evidence-based. Okay? So that's another reason. There's a third reason, at least I can think about. And we can go on and on till tomorrow. But anyway, let's think of a financial reason. If I have a written protocol, and in a minute I'm going to tell you it has to be registered on the clinical trial register, whether it's European or clinicaltrial.gov or whatever, then I may not want to invest time and money in doing a trial that's already being done in 3 other countries. I may not even get funded and put all the effort in it if it's already being done. It may help you modify your trial with pre-registered trial, okay? So there are a lot of reasons why you have to have a written protocol. I may have skipped some, but you'll get to know them when we write the protocol and go over them. It communicates the design and the organization. It writes in advance, and it's changed only in minor ways after the study begins. You can change the way you do your blood drawing. You can change the way you store your blood, and you have to document that. And it's a requirement for all regulatory pivotal trials that are going to get FDA approval. And interestingly, since 2005, the International Commission of Medical Journal Editors have mandated that they will not publish in the prestigious journal unless you are registered on a clinical trial register, okay, which is nice. And when I submitted to the New England Journal, even before they looked at my paper, they asked for the original protocol. They asked for the analysis protocol. It didn't get accepted, unfortunately, because it was a negative study, although it was powered to do so. And they asked for the post-protocol change in the analysis plan, if there were any. And Dr. Mahfouz, who helped me with that, provided me with some information. So be organized, save everything, date everything, because it needs to be handy on your hand when you submit protocols. And most big journals do that. And now, a clinical trial protocol by itself can be a paper. Marlene Schachtura, who wrote her clinical trial, Dr. Mahfouz is our statistician on it, for the 3-to-1A, it got funded. It's a published trial protocol on vitamin D in pregnancy. And it has an impact factor, and it's beautiful. Every time somebody tells me, tell me about your trial, I send them the BMJ paper online, the trial protocol. It's beautiful. It works for everybody. So I encourage you to think of that. So Spirit 201, Spirit document is followed in 201 and contains the following elements: executive summary, background and rationale with good quotation of the literature, study objectives and endpoints, study design, namely, what are the eligibility criteria, what is the baseline assessment, are these blinded, what are the endpoints, what's the recruitment like, who are you going to recruit, from where, how do you randomize, how is the sequence generated, what are your intervention, any other variable. Study procedures, if I'm drawing blood, I, in addition to the protocol, have SOPs booklet that tell you exactly where and how you draw the blood. Think of it this way. If you have 5 people on your team who are research assistant or fellows doing the trial protocol, all of them, right, going back to you for internal validity, should be doing exactly the same thing. So you have to have standard operating. How do I draw my blood? How do I get my bone density? How do I position the patient? Where do I store my blood? What temperature? Be picky, be compulsive. It's good for the trial protocol, okay? So all this has to be written down. Monitoring of safety and adverse event. You have a sheet for each visit, and you have, if you're doing telephone follow-up every 2 weeks, you have sheets for safety. We have adverse events and serious adverse events. What is the definition of adverse events? Very quickly. Something bad happens, right? Well, it's anything that happens after the trial starts. So what do you think the frequency of adverse events is in a placebo arm? What do you think? 30%, 50%, 80%? 30? Anything that happens after a trial starts. It's very often 80. Do you know what it is in the intervention? If it's a safe drug, probably 80. So adverse events are common. What's uncommon are the serious, but you have to document them. An adverse event doesn't mean it's the drug. It just means something happened after the trial started. I had a stomach ache. I had a headache. The patient had vaginal bleeding. The patient had, okay. A serious adverse event is what? Serious, so it must be serious. Anaphylactic shock? I like that, but why did you pick anaphylactic shock, not a heart attack, not a...
Mortality is high.
Okay, so mortality is one of the definition of serious adverse event, not for anaphylactic shock. So serious adverse events is any adverse events that results in death. What else? Disability.
It's life-threatening things, like threatening your life.
Okay, so death-threatening is not something I can measure. So it has to be death. It has to be disability. And one more, hospitalization. Serious adverse events, of course, are 2 to 3%. And they are to be compared between placebo and control. And serious adverse events have to be reported to your RRB. Now, data collection, how am I collect my data? If I say heart attacks, what do I mean by heart attack? Is it going to be documented by enzymes? Is it going to be adjudicated, confirmed? If I say hypertension, well, what do I mean by hypertension? How am I measuring my blood pressure? Triplicates, duplicates, 24-hour monitoring? Think about these issues. What's the quality control you're doing? Quality control is somebody coming and saying not only how you're collecting data, but how you're documenting data. Every single week, with every trial we've done, we have a lab meeting where we sit and review trial progress, case report forms. I have to sign. Dr. Nassar may have to sign half and me half. Quality control is very important. Dr. Mahfouz will tell you a lot about it, but very briefly, what's your design? Is it a crossover? Is it a factorial? Is it a typical one-on-one, two-on-one randomization? What are your endpoints? Hypothesis, data analysis plan, sample size? What's your power? Monitoring, interim analyses for something bad happening, and usually they are external boards called the Data Safety Monitoring Board that looks in an unblinded fashion at the results, events in the placebo and the control. And that's exactly what happened with WHI. After 3.5 years, they saw a signal, and they debated whether to tell the patient. They ended up telling them, but decided not to stop. And I think you will hear more, of course, from Dr. Mahfouz about stopping rule. When do I stop a trial? When I cross certain boundaries, by what definition, by what statistical test, etc. And if you do interim analysis, you have to adjust your p-value, right? Because you're already doing something that affects the p-value. Ethical consideration we talked about. So when I have to decide on a trial protocol, I have to first think and define my research question. And as I said, my research question may change as I look at the literature, as I discuss with colleagues, as I talk to my statistician, as I look at feasibility and cost. All of this enters the equation. Think through the science, do a very thorough literature search, and have content experts with you. No matter how much you read, especially if you're a junior, but even us as senior, it always helps to have several minds get together. Produce an outline describing the elements of the study. The study protocol very often is going to be around 25 pages, okay? And the IRB has to review it in addition to your IRB application. And develop SOPs, operation manual, to guide implementation. So if one of your RAs is sick, the next one knows exactly what to do. Test tube, red, 10 cc's in the left upper corner of that closet, seriously. Then she can do it exactly the same. Seek feedback and advice at every stage of the development. And again, Dr. Ware pointed me out to the finer criteria for good research question. Finer is feasible, F for feasible. Adequate number of subjects, adequate expertise, affordable in time and money, manageable scope and scale. I in finer is interesting. N, novel, confirms, refutes, or extends previous finding, new finding. Ethical and relevant to the scientific knowledge, to the clinical, and to the health policy, and to future research. So the finer criteria: feasible, interesting, novel, ethical, relevant. Trial registration, as I said, became a must by the editors in 2005. The National Library of Medicine, and some of us were lucky enough to visit that place. It's an amazing place in Bethesda, Maryland. It was established in 1997, and it requires registration of all trials, certainly, that are funded by the NIH, but many other people. I'm not funded for the pregnancy trial by the NIH. I'm partially funded now, but I have it registered there. It's a very recognized registries. And in 2012, again, there were over 100,000 trials in 50 countries. In 50 were from the US and many from other countries. So what are the ethical issues and at stake with clinical trials? I'm going to brush over that. Some of you, of course, have taken some of this, but a lot of organizations are interested in ethics in clinical trials. Of course, the WHO, there are GCP guidelines. There is the Office of Human Research Protection in the states, which oversees all clinical research across the state. It's very powerful. And if somebody gets a complaint, a blow whistle, from somebody who's worried about the way a trial is done, or even any NIH-funded study is done, there's an independent panel that reviews it. And there have been instances where OHRP has actually stopped, cold turkey, all trials, one point in famous institution. One of them, if we have time later, we'll talk about it, is the Hopkins trial on respiratory diseases. All NIH funding for Hopkins was stopped one day, just like this, stop. And it lasted, I think, a few weeks before they got their act together, revamped the way they reviewed clinical trial. And very briefly, it was an employee who was recruited for a study that looked at a drug that helps respiratory diseases, developed a cough, I forgot her name now, died in the ICU. And apparently what happened is the physician did not read the investigator brochure. The physician had a case of a cough on a previous patient, but didn't make the association. The investigator did not report the cough to the IRB in due time, nothing that would have changed this patient's course. And there was also conflict of interest because the patient is an employee and was asked to participate in the trial by Hopkins, and this is this is an NIH funding Hopkins, and she's vulnerable. She would be at hard time, she would be having a hard time saying no to enroll in the trial. And she thought that this is actually an FDA-approved drug, and it's not, so on and so forth. So it's a long story. Okay, so when do the ethics go back to? The ethics go back to many, many years ago. Of course, you probably know about the Nazi trials. So the Nuremberg Code is one of the earliest ethical codes. It says that informed consent is essential. Research should be based on prior animal work. You shouldn't just do a trial in human if there is no animal work. The risk should be justified by the results. Only quality, experienced scientists must conduct research. Physical and mental suffering should be avoided. And research in which death or disability injury is expected should not be conducted. And so the Belmont Report in 1979 was actually a report that came out of the US government outlining the ethical principles and guidelines for research in human subject, and the three pillars are respect, beneficence, and justice. Okay, and I won't review those in details. So whose role is it to protect human subjects? Don't look. Who should be protecting human subjects? You are going to do a trial. Who do you think is who are the people? It's a community who's going to protect research subjects. Who, guys, do you think is going to be the front line? I told you about the hip trial with calcitonin. I wouldn't do it. It's you. It's you saying, by the way, may sound cheesy, but this is how I practice. If you're going to do it to others, you have to accept it be done to you. Whether it's social ethics, professional ethics, clinical trial ethics, seriously. If it's good enough for you, then it's good enough for the others. So you are the first line ethical barrier to non-ethical research. The next one is the IRB. The next one is the institution. So when I was associate dean, I established something called the Human Research Protection Program, which is under the jurisdiction of the provost. And so if anything is NIH funded and goes wrong, NIH will talk to the provost, because we have a federal-wide assurance as an American institution telling them what are the ethical principles we follow at AUB. So the next one at AUB is the provost. In other institutions, they don't have that. They don't have an OHRP office, so there is a director and then there is a provost. And then it must be the hospital director or something, right? Okay. In the US, it's the provost of the institution, and then above that is OHRP, Office of Human Research Protection, which is this body, this ethical body that oversees everybody. And they also have a related office called Office of Research Integrity. Okay, so we started with the investigator, the IRB, the institution, but of course the sponsor, who's paying for the drugs. So at the NIH, the sponsor is the NIH, right? But drug trial, the sponsor is responsible. Pharma is responsible. Organization, you, society, brothers, sisters, husbands of potential subjects are. Okay. So the AUB-IRB mission, which I contributed to when I was associate dean in writing the HRPP governance, it's on the website. If you go on the provost website and you go HRPP or you go IRB, you'll find them there. So it's protect the right and welfare and privacy of individuals participating in research at AUB. Now, do you think the IRB should be doing just ethical review or scientific review?
Both.
Why?
Because like unsound or unscientific,
Excuse me?
fallacious protocol can lead to fallacious inferences and it can be unethical.
Excellent.
So it is the mandate of the ethical committee to look into both scientific and like the technical one and ethical.
So I like that. I agree with you. And this is how it was written. By the way, I don't know what it is now on the website. Maybe the sharp guys can go on the HRPP and see if the mission is changed or unchanged. But I felt very strongly, as you did, if it's not good science, it's unethical in multiple ways. For example, if it's a trial that has been done, it's not ethical to do it. So science has been done. Somebody needs to look at it.
People who are opposing this.
I know. We're getting to that. Excellent. So you're hot on the topic. So if it's not scientific, it's not ethical. If the protocol design is not correct, the conclusions are not correct, then the practice is going to be incorrect and it's unethical. But if you look at the IRB, at least when I looked at it back then, and there was a lot of struggle on campus, people hated me when I put it together with the provost as such. This is why I want to know if it's still this way or change. They said, that's none of your business. We are the investigator. We know the science. The IRB should just review the ethics. And for me, they're very closely intertwined. And when we looked at the landscape in the US, actually 40 to 50% of IRB were science and ethics, because non-scientific projects for me are unethical, but 50% was just ethics. So I don't know what it is now in the States. I should have reviewed it, if it's a 50-50 or 70-30. And I would love you to tell me if it's unchanged on the website, okay? But I think I agree. I think, anybody wants to say anything else? The question is, does the IRB have the scientific expertise to give the science feedback?
I think even if the IRB does not have the scientific expertise, you can still call for experts to come and review.
Love it. Excellent.
You can give suggestions to the scientific committee if there is a separate.
So you do get your experience, because I still think it's your duty to make sure it's scientifically sound, to expose your subject to no matter how little risk you do. And I think at AUB, a lot of research is funded by AUB. I don't want somebody to do a study that's nonsense and take the resources from MPP or the NIH or whatever foundation to do something that's not going to contribute to the knowledge and that's not sound and ethical. So I have strong feelings about this. That's not shared widely. Okay, but I agree. You can get the scientific expertise, and that's what the IRB does. All right. Anybody else wants to say anything about this? This is a very debatable issue. It's very hotly debated in the States. I think the dust settles. Most universities in the States now do both. But back then, 2009, it was kind of a split jury on that. Okay. So the IRB provides professional guidance and support to the research community and complies with the Belmont Report, institutional policies, and federal law as applicable. The World Medical Association Declaration of Helsinki originally started, has had many declarations. This is just their website, and you can see how many of them they have, and all of them had to do with the ethics and revisions of the ethics of research. This is the controversial placebo issue, right? We mentioned that. So the ethics of placebo-controlled trial, the WMA came on that, and it says the benefits, risk, burdens, and effectiveness of an intervention must be tested against those of the best current proven intervention, except it gave an exception, because when it across the board first said it has to be against an established intervention, you can see there were a lot of issues there, because in some instances you really need a placebo-controlled trial. So they made an exception, and they said except when the use of placebo, no treatment is acceptable in studies where no current proven intervention exists, or where, this is how they got away, giving some leeway to the ethical board and the investigators to manage this issue, where for compelling and scientifically sound methodologic reasons, the use of the placebo is necessary to determine the efficacy and safety of an intervention. And the patients who receive placebo or no treatment will not be subject to any risk that is serious or irreversible harm. Extreme care must be taken to avoid abuse of that option. So this is the loophole. This is the option that they gave after the original declaration to allow that. And the other thing, this is the original declaration. In any medical study, every patient, including those in a control group, if any, should be assured of the best proven diagnostic and therapeutic method. So this was the original one that led to the controversies. So what are the arguments? There have been many papers discussing the ethical basis for placebo-controlled trials. This is the against argument. These are some people, this is a paper many years ago. Providers should always act in the best interest of the patient. There is no circumstance in which an effective treatment should be withheld. Placebo-controlled trials are unnecessary. They test only significant differences from placebo, not improvement over baseline, which is the current state of management. So what do these people suggest? And I don't know, Ziad, you're going to discuss that very briefly, right? Non-inferiority trial. So they actually say that active control equivalence trial, non-inferiority trial, or ASSET are the alternative. Patients decide to a new treatment or to another treatment believed to be effective, and a null result suggests that the new treatment works because it's not inferior to the known intervention. The problem, the sample size becomes big. It violates the principle that trials should be as small as possible to minimize risk. The meaning of a null hypothesis, no incentive to run a clean trial, it becomes more murky. The poorer the trial potentially, the more error variance and the higher the probability of a null result. So if it's a poorly conducted trial, you're likely to come up with a null result. And so this is why the design, the power should be predefined and well determined. When we submitted to the New England Journal, we had 3 reviewers. 2 were excellent. 1 was really harsh on us. 1, he had a problem not having a placebo. And the reason we didn't have a placebo, it was a vitamin D trial in elderly, and there is a lot of hypovitaminosis D in Lebanon, and the concern is it could lead to fracture, hyperpara, and osteoporosis. We didn't feel comfortable to have a placebo. So we had the Institute of Medicine recommended dose, the low dose of 600 versus high dose. So one of his harsh comments was, this is not a placebo-controlled trial. Well, that's not a comment I can do anything about. The study is done, the trial is gone, the design was done, full stop. And then he said, it's a null result, so it was not powered. And he did not read the method because we were perfectly powered. So unfortunately, sometimes you get hit harsh by a harsh, if he wasn't for him, I'm almost sure he would have done it, made it to the New England Journal, but it didn't. So it goes back, if it's a poorly done trial, if it's not adequately powered, a null result mean nothing, right? So I will try to attend the lecture by Dr. Mahfouz because I understand these non-inferiority trials, honestly, the least. This is now placebo pro argument. Placebo controls are an important part of medicine's commitment to ensuring the safety and efficacy of new drugs are established solidly. Informed consent, saying that you're going to use placebo in the informed consent, cannot be invoked to justify an inappropriate trial design. In other words, this is what, you cannot say, it's okay, the patient signed for placebo, and so I'm going to use a placebo trial as my argument because they signed. This cannot be adequately accepted. This is now, again, on informed consent. Investigator, the title should be changed to on informed consent. Investigator must be extremely thorough through obtaining consent from patients. And I will always quote this, maybe some of you have heard me, an instance where we're doing, it wasn't a trial, we were doing genetic study on a family from Syria where they had a very rare disease called MEN. The father had severe hypercalcemia, kidney stone, renal failure, was on dialysis. He had a non-related wife and children, and the question is, did they carry the MEN gene? And the people who did that were at the NIH. Now, just to do the genetic study, because genetics brings a whole dimension to ethics, right? Who is the father? Who is the mother? Do they have something deadly we could discover while looking for a specific gene, etc.? Because it was a genetic study, the NIH developed a 7-page English protocol translated to Arabic by their lawyer, back translated to English. I was on the line with the family in my office, the lawyer at the NIH, the investigator colleague who's doing the genetic study, and me, honest to God, almost 2 hours, maybe a bit more, going word for word over the consent form. So giving the point is, and it was worth every moment, because it turned out the kids didn't have the genetic mutation, and it was a very nice effort and time investment. But anyway, you cannot hand the patient the informed, and this wasn't a trial, you cannot hand the patient a consent form and say, read it, if you have any question, let me know, he signs and you're happy. No. And some patients will tell you, because the education level is not as developed, they will tell you, I trust you, doctor. You're my doctor. You decide, I'll sign anywhere. Where do I sign? No, you should not accept that. If they don't accept to go word for word over it, at least explain in detail what it is. Give them a copy of the consent form to take home, even after they sign. That's a requirement from the IRB. Why? Because they can share it with more educated people, they can share it with spouses, they can have second thoughts. And so you want them to know exactly what's going on. Sometimes consent forms become scary because they become so long, and the IRB, who's on the patient side, may put a lot more there that's so scary they don't want to do it. But you have to live with it. You have to do it. And the negotiation is, do we need to put all the detail? I think all the salient details have to be put, the risks as they are, the potential benefits, and don't make it sound better than it is. Okay, so this is a slide here. I've jumped through slides illustrating from 1977 to 2002 the average page length of the consent form, which was 2 pages in 77, and it's 5 pages in 2002. I can tell you it can be easily 7 now. And versus the percent of risk discrepancies over time, the discrepancy between the actual risk from the trial and what's written, it was very high. And now it's very low. So you're asked to do more, to give more details to the patient about the trial, and it takes longer. So this is like the patient I told you. Here is a mouse saying, do you really think we need to sign this? And this one says, oh yeah, I already did. Patients should take the time to read the clinical consent form. And the other thing is, you as a physician, this physician who is still wearing the white blouse, saying to the patient, I want you to concentrate here, consent, information, comprehension, voluntariness. And sometimes patients get paid for participating in trial, and this patient is drooling because he wants the reward from participating in the trial. The other thing that you really have to be very careful about is the perception from patients that once they're in the clinical trial, they're getting drug therapy, like the case at the Hopkins. They need to know it's an experiment. They need to know you're not guaranteeing them benefit, but they do get some benefit, unfortunately, in our part of the world where they get free physical exam, lab studies, copies of lab studies. It's interesting because when I was in Boston, my patients in my trials were all PhD educated people. They want to advance science. When I came to Lebanon, my patients were mostly non-insured people who want to get free access to physical exam. But we do go out of our way in the elderly trial. If we discover hypertension, we make an appointment in the OPD. We walk them to the OPD because it's cheaper. We try to explain to them. So we try to really give them the benefit of being in a trial. But you have, this is just to remind you to do that. Okay, Another layer of complexity, some of it I touched upon when I explained the elderly trial here, is special ethical considerations in developing country. Many editorials on this. I will tell you one of them. We did a cross-sectional study 5 years ago where we wanted to look at bone density in older Lebanese subjects. We don't know what it is. So we had to bring older subjects to AUB. One day, blood test, bone density. And I had a nightmare. What's my nightmare? I think I told you, you sharp students or not. What's my nightmare bringing elderly subject to AUB? No. What could happen to them coming to AUB? What do we do very well at AUB? Or in other clean places? What do? We clean the floors, wet floors, caution, slipping and fracturing a hip. Now, why is that a nightmare? A lot of these elderly people are uninsured. Who's going to pay for the hip fracture? Who brought them to AUB? So what did we do? We were lucky because it's a one-day exposure. I spoke with risk management at AUB. This is a real-life story. I'm not making it up. And I said, I'm bringing these elderly subjects. If somebody slips, I want to be able to give them care. I don't have money in my grant for care. Can you cover it under risk management? She goes, this is what you're going to do. And they could. It's becoming more difficult now because there are more investigators doing trials. This is what we're going to do. We're going to have you send us, every day before the day they're coming, their list. So they will be covered for that day if something happens, but not for the whole year. It was perfect, and that's how we did it. So think of ways. Now, if I was doing a cancer trial giving intra-arterial chemo and the artery blows, the AUB is not going to pay for it, of course. So it's becoming more complicated. But always think of these ethical issues. You have a vulnerable population, more vulnerable even in developing countries. The other issues of research in developing countries, HIV trials in Africa. Okay, they're developing. Why in Africa? Well, Africans can't afford. Many of them are drug naive. This is where it started. But what's the danger? After this drug trial, using this wonderful population, efficacy gets established. What happens? It will be used in the US and Europe, but the Africans cannot afford it. So one of the ethical requirements, and has been proposed by certain organizations, we'll find it, is providing free drug or providing drug at a very reduced rate for these populations where you developed it. It has to be, one of the ethical considerations in developing country, the drug has to be relevant to the population, accessible to the population after trial completion. And I can go on and on and on, but I'm not going to go on and on and on. Tuberculosis, same thing. A lot of guidelines that offer suggestions for safeguards, they are variably followed in different places. Oh, one of the way pharma actually safeguards the right of the vulnerable population in the Middle East is they give them, the IRB at AUB would not approve the drug trial unless they give an insurance for the trial for all study participants while in the trial. That's good. They don't do that in the States. But there's always the responsibility of the physician to try to defend the patient. For example, let's say you have a patient who has a peptic ulcer disease, and you know that from the baseline information sheet that you filled, right? And let's say you're using a hypertension trial, and the patient bleeds from the ulcer. Are you going to cover him under that insurance, or are you not? This is where the discussion between the IRB, the physician, and pharma goes on. And this is where when you fill, and I didn't tell you all that, you will get it in the clinical trial protocol, when you file an adverse event, for example, gastric ulcer bleeds or ulcer bleeds, and you try to get as much information, what did endoscopy show, etc. You say, you have a menu of options. Is this drug related? Yes, no. And for yes, it has multiple menus. Possibly related, unlikely to be related, highly likely to be related, and same thing for the no. So you try to put some strengths of the evidence to the association between the adverse event and the study drug that's being used. But if the drug was aspirin and the patient has an ulcer, pharma may say he had an ulcer. I may say, well, but he's trying your drug, so we need to cover. Very often, I have to say, pharma has been very positive about covering, even when in doubt, that there is a drug relationship to the adverse event. So that's one of the safety mechanisms we have in developing countries. They don't have in the developed world. Well, actually, when I used to make signed patients' consent form in the States, they say you are a third-party payer or you may be billed for any adverse events that can happen during the study. In Lebanon, you have another problem. If it's an investigator-initiated trial, yani there is no pharma, there is no big pockets to cover. You are the investigator, you got $40,000 from MPP to do your drug. Who's going to cover the AE? That's the problem. Consent forms currently say if you develop an AE, we will not be covering for that AE because we don't have money. Right now, the government, so this is how government gets into ethics. The government has stipulated that everyone doing a drug trial has to have some insurance coverage for the patients. So AUB now has a funding mechanism where to not stifle science and research, they actually committed to being responsible for paying for AEs if they happen to patients. Now, I have a problem with that because if the patient's AE is unrelated, and this is where I had a discussion with Walid Ammar, I said if it's unrelated, I really don't think AUB should be paying. And he said, well, AUB can afford treating poor patients. And it's an ethical discussion. But ultimately, they left it kind of a little bit flexible so that you could have some leeway into deciding as an institution what to cover. But at least it's requiring the coverage by the institution for investigator-initiated trial. Now, here is some of the safeguards. I'm highlighting the important one, Helsinki Declaration. At the conclusion of the study, every patient entered into the study should be assured of access to the best prophylactic diagnostic and therapeutic methods identified by the study. Wow, that is very ambitious. But that's what we're getting at. Providing at least free drug at the intervention completion is important. So there are other documents that called for similar provision from the Geneva Joint United Nations Program on HIV, ethical consideration HIV preventive vaccine research, NIAID's guidance document stipulates the same thing, so on and so forth. So ethics could be a course by itself. We're not doing that. I'll be happy to discuss things with you. I have a lot of backup material. Dr. Arawi has a lot. And we have some online resources. Now, some of the regulatory aspects, because drug trials are regulated. They're regulated by the funding agency. They're regulated by the ethical body that oversees what you're doing, and so on and so forth. So few regulatory comments to complete. This is the clinicaltrial.gov website. I would invite you to just click on it and try to see what it has. This is our study posted many years ago. It's published now. We have 2 publications. And we finished on time. So we have one in American Journal of Clinical Nutrition, another one in the JBMR, Journal of Bone Mineral Research. But we have many other on secondary and exploratory outcomes that are hoping to be published. So it's all in there. It has all the details. So please click on that. Now, the FDA has a lot of regulatory CFR documents online. I'm going to read what the subjects are. So we have electronic records. What's the rule for electronic signature? What's the informed consent guidance? What are the safeguards for children in clinical investigation of the FDA? What are the consent form elements stipulated in CFR 50? What is the exception for general requirements in an informed consent? Financial disclosure. If I'm getting paid by pharma to do the drug trial, I should say that in the informed consent. This is there. IRBs, how they review. FDA IRB registration rule. Our IRB is registered with OHRP because it's an American institution. Good laboratory practice for non-clinical studies. Investigational new drug application. If you have a drug you want to investigate outside pharma. Foreign clinical trials not conducted under, and they're on and on document. I'm not going to go over them. So by the time you finish writing your serious protocol, you will have a pile of paper piling up and piled up that you have to sign. And this one has huge piles of papers. And the RA, I guess, is the one who is sweating, and the PI or the IRB or whoever is the authority there saying, and this is the nth form, is the form you send back to confirm you send back all the forms. So there is a lot of paperwork to be done. I'm going to conclude with negative trial. There is a lot of publication bias. From journals, they only want to see positive trials published, which is why we didn't get published, I think, in the New England Journal. That's bad, but I think it's important to carry that message. But of course, this is if it's a well-designed negative trial. And this slides from a recent paper illustrate the likelihood of null effect, finding no positive effect, of large NHLBI trials, and it has increased over time, okay? So null effects are becoming very common with big trials that are appropriately designed. So people are accepting null as something we need to know about so we don't invest in it. These are two analyses on the primary outcome. In NHLBI is one of the major funding institutes in the States. And this is showing you a meta-analysis of drug and supplement RCT primary outcomes published before 2000. You can see that the effect size here is significant. And you can see that in a more recent publication over 2000, they are more likely to cross the confidence interval. And it's just telling you people are more accepting of negative trials, provided they are well designed. Compliance with results reporting a clinicaltrial.gov registry. If you look at industry, it's very highly compliant. Why? Because they have something at stake. They have to have a product approved. The NIH comes next. The percentage of trials sticking to the regulation are 40%. For industry may reach 40 plus. But others, if it's not NIH, it's not industry, the compliance with reporting results on clinical, this is reporting results on clinical trial. And I have to tell you, I have not, I am one of the culprits, reported our results of the elderly trial on clinicaltrials.gov. Can you guess why? I mean, it sounds great reporting everything on the clinical trials. Can you just, okay, just brainstorm about why I didn't, although I think I'm a serious committed investigator. Why do you want to publish on clinicaltrials.gov? I've already published, I told you, in 2 major journals, prestigious. I'm very happy. Why do I want to publish on clinical trials? Why would I be wanted to publish on clinicaltrials.gov?
Why do you want to publish exactly? Because it's a positive result. Many people prefer publishing positive results.
Okay, but if it's positive and it's a solid trial, I should get it in the literature. So it's actually the other side. They want you to put your results on clinicaltrials.gov because they want to be able to see negative trials in case they're not published. Well, they don't get published because journals don't like negative trials, although I think with time they're liking them more and more, as I've shown you. But negative trial results need to be registered somewhere. You need to be able to see that. And I think this is important. I didn't publish mine because I already published, although the rule is for me to go back and publish, but because the way they want you to publish your results, if you go, is a whole new different format. It's not like you put your manuscript or you put more details or analysis. They have specific ways they want you to do it, and it's very time consuming. And I don't have time to even publish the next few papers and write them up decently. So that's honestly the reason why I didn't. And I feel bad, I'm not proud. But there are practicalities. يعني مثل ما بيقولوا اقرأ تفرح جرب تحزن. You know, you read, it's really wonderful, I'm going to do it all the best. But in reality, there are other things that pull you in direction. I think negative trials should be published. I really do. I think there should be maybe a stipulation only for negative trials, because if positive results are published somewhere, maybe that's good. But it's very time consuming. You need resources that you may not have. I was at the NIAMS, it's the National Institute of Arthritis and Musculoskeletal Disorder, public hearing board meeting 3 years ago, and there was a whole session dedicated there on all NIH-funded studies should have a data repository with the results on negative trials. And I thought that was wonderful. I don't know whether they have reinforced that, but that's telling you the direction of where science is going. Okay. This is again media, spilling the beans, failure to publish results of all clinical trials is skewing medical science. You're only hearing about positive studies. I like this. I've always used it as my last slide. I have a couple more that are more, kind of permutations on a few issues I raised. But this is by the American Society of Bioethics and Humanity, a JAMA perspective. It's sometimes difficult in light of potential financial rewards or fame to instill in researchers sound moral thinking, allowing them to manage future controversies. Researchers should ask themselves, is it something good to be done, or are we just doing it to be first? So I think it's really neat. And this cartoon says, I can't believe schools are still teaching kids about the null hypothesis. I remember reading a big study that conclusively disproved it years ago. And so there is a series this year that I've shared with Dr. Mahfouz and Dr. Ail, who give clinical trials, who have the same interest. There are a series, they're called the Clinical Trials Series. I've outlined the references. The key ones, I think, to keep in mind is drug challenges for small biopharmaceutical companies. It takes billions to develop a drug, so some drugs may not come to the horizon. Mike Rosenblatt, you've seen some of his slides. This is when he was at Merck. He's now on his own. The large pharmaceutical perspective. Now, look at this. This is interesting. I've mentioned IRBs. I've mentioned the provost. I mentioned pharma. I've mentioned communities. This is, I think, a very telling cartoon. Factors contributing to the complexity in large clinical trials. First, the stakeholders, i.e. the patients' advocacy groups, the medical community, journals, and industry. The payers are contributing or pressuring in the direction of clinical trials. Insurance companies, national health agencies, patients and consumers. I told you, our Minister of Health now has a mandate, for example, for who covers side effects in clinical trials. Regulatory agencies. There is multiple independent agencies that have requirements. The FDA has its requirement. The NIH has its requirement. The EMA has different requirements. So it's really interesting. Geography is another factor. Multiple sites, multiple investigators, multiple IRBs. Some may approve one consent form, others do not. Variety of standard of care. What's accepted in Lebanon as standard of care may not be accepted in certain countries. Variation in ethnic backgrounds. Variation in diet. How does it interact with the drug you're using? Variations in alcohol and tobacco use. The most interesting thing to me is one of the most recent osteoporosis drug trial that was presented last year at the ASBMR. It's called romosozumab, which is a sclerostin antibodies that increases bone formation and decreases bone resorption, showed results very different in the US and the rest of the world from Latin America. Interesting. And some people started scratching their head. Why would that be? The results in Latin America showed no reduction in fractures. Can you think of a reason why?
Dietary factors? Dietary factors?
No, it's a fracture trial. Let's dissect the problem. It's a fracture trial, so the outcome is fracture. Okay? The outcome is fracture.
Lifetime.
Okay, perfect. So what I'm getting at is that the number of events, the power of the study for Latin America, because incident fracture for lifestyle reasons, for dietary reasons, for exercise reasons, events in the placebo were much lower than in the rest of the world. And it's recognized, we've published on this, there are wide geographic variation in hip fracture. It can go up by 20-fold from Japan and China to the Northern European countries. So when you take Latin America, we are actually like Latin America and Lebanon. Our fracture rate is lower. It's more like Southern Europe. It's certainly lower than the States. So when you take a low baseline, I mean, it's an explanation. We think that's what it is, but we don't know for sure. So everybody said, oh, but Latin America responded differently. Interesting. So maybe that's what it means is, by the way, if the sample size was not enough in Latin America, could you prove drug efficacy in Latin America? I have a public health teacher, Ziad, please correct me if I'm wrong, who tells me I can always find a significant result if I increase the sample size. But the question is that effect size clinically significant? I can make the study 5 times as bigger and show fracture risk reduction in Latin America, but it could be that my relative risk reduction is only 10% compared to 40% in the US. Is this a fair statement, Ziad? So you can always find a significant result, yeah, but is it really? This is where you need to think as clinician and public health official. Is that difference you're going to find really clinically or public health or whatever your perspective is relevant? Okay? So geography is important, yes, possibly, for multiple reasons. Maybe the disease you're studying in that country is not relevant in other countries. Design, primary and secondary endpoint, composite endpoint, surrogate markers, biomarkers. So it's not just one endpoint. It's not as simple. Personnel, look at how many people are involved. Investigator, the trial monitor from the drug pharma who comes and checks your case report form, the biostatistician, the project manager, data safety monitoring committees, academic collaborators, consultant, and on. So it's become a very complex business. And because it's complex and because you want to squeeze the trial and get as much as you can out of it, and because it's very costly, this last perspective in the New England Journal compares in 2002 some trial design characteristic with 2012. And I'm just going to read them. So in the earlier years, the number of endpoints was 7. 2012 became 13. The total number of procedures is 106 in 202, 167 in 212. Total number of eligibility criteria was 31 in 202, 50 in 212. Total number of countries, 11 in 202, 34 in 212. Number of sites, 124 in 202, 196 in 212. Number of patients undergoing, this is on the average, randomization, 729, 597. Total number of data points collected not available then, 9,029,000 and 203. I think that's really telling. The last thing is, in this series, is patient-reported outcomes. So it's not the outcome that you register, but you also give weight to patient-reported outcomes because that matters. And I really think that title is very telling. Harnessing the patient's voices to improve clinical care. And I love that. So this New England Journal, again, paper looks on the y-axis at patients visiting emergency department in the usual care clinical trial versus the patient-oriented harnessed clinical trials. And you can see that there is less ER visits there because patients are inputting into how clinical trials are read. And the probability of overall survival is better, maybe by 5 to 7%, in the patient-reported outcomes in a cancer care center compared to the usual care. So I think it's really interesting because patients, subjects are integral to the success and to the operations of clinical trials. So I guess it's what time now? 5:20. So it's almost 2 hours. We went beyond one. I thought it was 1.5. I hope you got some flavor. This is really supposed to be more of a flavor, a journey of what clinical trials are all about from somebody who spent many years doing it and enjoying it. And I'm open to question now and later.
I have a question regarding the efficacy and effectiveness.
Yes.
In the real life, like the effectiveness decreases or it may decrease?
I think in general, the experience with impact of drug on an outcome is less in the real world for the reasons I mentioned. Because there is adherence, whether they take the drug, there is interaction with other drugs they may be taking, there is lifestyle modification that may be different from the trial, there is patient population characteristics. So in general, effectiveness is less than efficacy. Efficacy is the best outcome you can hope for. And by the way, one thing I didn't touch on, but Dr. Mahfouz will, is intent to treat versus per protocol analyses. I'm sorry. In a drug trial, the efficacy is much better if you do per protocol. In other words, you only analyze patients who took the drug as they were taking it, as opposed to taking the adherent and non-adherent and all these issues. So the drug trial is the perfect scenario. And effectiveness is less, in general. I can't think of an example where it's the opposite.
So you mean there are other factors that drag with backline? So like the effectiveness is decreased?
The efficacy is decreased, so the effectiveness is less. It's called effectiveness because it's in the real world. It's the same thing. Risk reduction, okay, is your measurement of drug efficacy in drug trials, okay, relative risk. In an observational study, because in the real world you're going to compare those treated to those not treated, but it's not a drug trial. So you're looking at risk reduction in a case control way, and it's called effectiveness. Okay. Any other questions? Okay. So the slides are on Moodle. The backup stuff, the PDF for the clinical trials that I presented are all there. Rima can answer questions. I'm available through Rima to make an appointment, and I look forward. I'll try to see you for some lectures Dr. Mahfouz will be giving, at least the one where I feel I can learn a lot. Crossover, factorial, negative, non-inferiority trials, and I'll be there the last day. Okay? All right, have a great weekend. Thank you for coming. We have a big class. We have more than I thought.
So I'll see you tomorrow at 9, all right? I'll be here to open the door for you.